As I mentioned in the previous video, the innate immune response is the general immune response that our body has to any sort of tissue damage or infection. This is our general response to what are called PAMPs and DAMPs. So PAMPs are pathogen-associated molecular patterns, while DAMPs are damage-associated molecular patterns. These are the responses that our cells produce when they either encounter an infection or they are damaged. The other thing a PAMP could be is a PAMP could be a molecule on the surface of a pathogen or a molecule produced by a pathogen. So not only can our body signal, hey, there's something wrong here, to recruit the immune system, but those infectious agents themselves can stimulate an immune response. Our first line of defense against any infection are the various physical and chemical barriers that we have. So shown in this image of a person, we can see some of the physical barriers, which include our skin, ciliated epithelium, but also some of the chemical barriers, like lysozyme, which is an enzyme that breaks down peptidoglycan, antimicrobial secretions, the acidity of the stomach, the production of bile, the acidity of urine. We have a number of different mechanisms to keep us from getting infected, to keep microorganisms from getting inside of our body and establishing themselves. So what I really want you to appreciate from all of these slides about the immune system is the idea that we are constantly exposed to microorganisms, and our body does a very good job at keeping us from having infections. And a lot of that is in response to our very successful first line of defense. In our innate immune response, some of our frontline molecules include antimicrobial peptides and ion chelators. And you might think, well, now what in the world does an ion chelator have anything to do with protecting us from infection? Well, in our bodies, metals and ions, especially things like iron, can be very limited, but are absolutely necessary. For many microorganisms, that is the case as well. So if our body has the ability to sequester those ions, it prevents the microbes from using them and prevents them from being able to grow. We have various antimicrobial peptides that are produced that can do things like break down peptidoglycan. I gave you the example of lysozyme. We have other peptides that are produced that can induce immune cell migration. We can disrupt microbial cell membranes. And again, this is just the general response to anything that's trying to get in. Another component of our immune system is our complement system. And I'm not going to ask you the details of how the complement system works, but there are 3 different pathways that kind of all end up with the same consequence. And so the consequence here is shown at the very, very far right. And it's the production of something called the membrane attack complex, or the MAC. And the idea is it's a collection of proteins that actually form a pore in the membrane of an invading, especially bacterium, that can lead to death of the cell. We can also sometimes have this happen on our cells, which is usually pretty bad. So we want to target this towards our invaders. So we have to be very careful about what activates this particular pathway. So there are 2 pathways that are antibody independent. So there's the alternative pathway, which is kind of just randomly on, but then when they encounter microbial surfaces, you get the eventual cascade that leads to the membrane attack complex. There's the lectin pathway, which recognizes carbohydrates that are generally only found on microbial surfaces. And then you have the classic pathway, which is activated in response to antibodies that bind to the pathogen. And antibodies are generally only present because they're synthesized by the adaptive or antigen-specific part of the immune system. So something you can, I guess, reflect on is, in your opinion, is the classical pathway part of the innate immune response, or is the classical pathway actually part of the antigen-specific response? Another part of our innate response is, of course, our interferons. These are cytokine-like proteins, and they interfere with viral replication. And the way they do that is that they can lead to the transcription of antiviral proteins. They can cause fever. When the temperature is too high, the cells can't reproduce. And they can also lead to the production of T cells. A healthy T cell response is really critical to fighting viral infection, because generally what we need to have happen is the destruction of virally infected cells. So interferons are critical in order to have a great viral response. We also have cellular components of our innate immune system. I showed you those on a diagram in the previous set of slides. So we have a group of cells called granulocytes that consists of neutrophils, and their I guess classification are here. They have numerous cell surface receptors which recognize microbes. They promote phagocytosis. They have granules inside of them with antimicrobial substances, and they can exocytose those in order to deal with pathogens. And neutrophils are actually a major component of pus. So when you have an infection that's called purulent, meaning it produces pus, neutrophils are a big part of that. Another group of granulocytes are the mast cells, basophils, and eosinophils. These are usually found at lower levels than something like a neutrophil. If you go back to the previous set of slides, you might remember the table where it showed how many of each cell are common in the body at any given time. But they are granulocytes because they do contain those granules with antimicrobial substances. They are potent inflammatory mediators. So these things really drive a generalized inflammatory response. And with an inflammatory response, generally what we're seeing is heat, and that can be localized heat, or it could be a full body fever, pain, generally at the site, redness, and swelling. And the redness and swelling generally come from vasodilation. So the blood vessels kind of open up to allow these cells to get to the site of infection. Well, that can also bring out red blood cells, which leads to the redness and the fluids that are contained within those blood vessels, and that can cause the swelling. The pain is the release of a small protein, the bradykinins, that can activate that pain response. Now, mast cells and basophils are responsible for releasing histamines and cytokines. So you actually may not like these very much if you get allergies. These are also part of the allergy response. So are eosinophils, but eosinophils are also really important because they are antiparasitic. They are one of the few cells of the immune system that are designed to help protect us from parasites. So in the US, we don't generally see high eosinophil levels because we don't generally get a ton of parasitic infections. But in allergies, sometimes we get them. So if you have people with eosinophilia, sometimes they get significant allergy symptoms. We also have cells of the Monocyte-Macrophage lineage. These are really important because they are long-lived phagocytic cells. And phagocytosis is really important, not just as a mechanism of pathogen elimination. Yeah, sure, phagocytic cells go around and gulp up all of these pathogens and digest them and get rid of them. But really, one of the critical features of that is not just that they gulp them up and digest them, but then they show those pieces those antigens on the surface of the cell. So they are called antigen presenting cells. And macrophages can be divided into two different lineages, M1 and M2. Again, this is not an immunology class, so I'm not necessarily going to be asking you tons and tons of details about the differences of all these kinds of cells, but I do think it's important that you're aware of what our immune response is. So the Th2 activation, this is a type of T cell of macrophages, leads to M2 macrophages. And for the most part, M2 macrophages are more important when there's damage. So they are really important in tissue repair. On the other hand, they also can promote tumors because they do things like stimulate angiogenesis, stimulate fibroblasts, etc. The M1, I guess you could say, are, more aggressive towards infection. They drive things like inflammation and fever. They have strong antimicrobial activity. They also have anti-tumor activity. And you might think, well, why in the world would we keep cells around that promote tumors? But their main function is really tissue repair. And if we don't have tissue repair, how do you heal a wound? So we have to have a balance of both of these cell types in order to really have the best and most robust immune response. Dendritic cells, they are like the master. They are sometimes called professional antigen presenting cells. Like that is their job, and they are considered to be, as you see in this bullet point here, the bridge between innate and antigen-specific immune responses. They have all of these tendrils, and you can see they use these tendrils to grab and phagocytize various antigens. These are conidia. They're a type of reproductive spore that various fungi can produce, but you see this cell is just like grabbing them, bringing them in. And then the dendritic cell will present the antigens that it phagocytizes to various cells of the immune system, like CD8 positive T cells or CD4 positive T cells. In a different type of dendritic cell, a follicular dendritic cell, these are in those B cell areas of the lymphoid tissues, like those germinal centers I pointed out in the spleen or in the lymph nodes. These dendritic cells will present to B cells. They'll present antigen to B cells, and that is what eventually will allow things like antibody production and differentiation into plasma cells.